Hi, I'm Tan Zhengqi. In this video, I'm going to explain about the gene regulatory mechanism of mammalian cells in response to heat stress. Heat stress is mainly caused by the imbalance of heat production and heat dissipation in mammalian cells, often due to the elevating environmental temperature and humidity. This imbalance can cause the cellular protein to be misfolded or denatured, and prolonged exposure to heat stress will cause mammalian cells to damage. Therefore, heat-responsive genes in the cell are turned on by the binding of HSF, heat shock transcription factor, to the heat shock promoter of the gene to produce heat shock protein that protect the cell from damage. Let us start off with the non-stress condition. In the non-stress condition, HSF is remain bound to heat shock protein 70 and 40 as an inactive monomeric form in cytoplasm. During the normal condition, RNA polymerase and the initiation factor GAGA already bound to the heat shock promoter of the gene. GAGA initiation factor will recruit the co-activators, general transcription factors, and nucleosome remodeling factors to form a pre-initiation complex. CDK7, which is a part of TF2H, the transcription factor, will phosphorylate serine 5 of the RNA polymerase tail to initiate the transcription of the first 20 to 40 base of the gene, 20 to 40 bases of the gene. When there is no heat stress, the RNA polymerase will stop at the base number 40 by the negative elongation factor and DSIF, which is composed of SPT4 and SPT5. Only upon heat shock activation, HSF can dissociate from the protein chaperone and homotrimerize itself, forming a trimer. And this trimer will be transported into the nucleus for post-translational modification. This figure on the left will show the signal transduction pathway of the heat stress signal to activate the gene transcription of heat shock protein. So the pathway is known as the SAPK pathway, stress-activated protein kinase pathway. The pathway starts off with the heat stress signal detected by the membrane stress receptor to induce the activation of MAP3K proteins. That is, particularly for heat stress, will be ASK1 and MEKK1. These two proteins will then activate MAP2K protein, that is MAP2K3 and 6. Finally, this MAP2K protein will activate P38 MAPK protein. P38 protein will phosphorylate heat shock transcription factor at post-translational level so that the phosphorylated heat shock transcription factor can bind to the heat shock promoter. When the HSF bound on to the heat shock promoter, it will recruit additional co-activators such as PTAP and nucleosome remodeling factors. PTAP will phosphorylate three things. First, the serine 2 of the RNA polymerase tail. Second, NELF, negative elongation factor. Third, SPT5 of the DSIF molecule. The phosphorylation reaction will cause the dissociation of the negative elongation factor from the RNA polymerase. Now, RNA polymerase is released from its paused state to productive transcription elongation, lead to the production of heat shock protein that repairs and stabilizes protein that is affected by the heat. That's all. Thank you.